Okay, we're going to do a quick review for your first Case 21 test, and we're going to start with characteristics of life, and we went through a good many characteristics of living things, and the first one, of course, is that all living things have cells, and of course, you can have one cell, you can have many cells. When we look at types of cells that organisms have, prokaryotic cells do not have organelles. They do not have membrane-bound organelles. We call them bacteria, but remember they do have ribosomes, which you can see down here in this picture. All eukaryotic cells have membrane-bound organelles. They have a nucleus. They have their DNA organized in chromosomes inside the nucleus, and so remember they're just a lot more complex. All living things reproduce, so make sure you know the difference in sexual and asexual reproduction. Of course, sexual reproduction means you have two parents typically, and asexual means you have one parent. All living things have DNA, so even bacteria have DNA. Their DNA is just circular. It's not organized in a chromosome. Living things grow and develop, and we'll talk about organization in just a second. Make and use energy, so remember that organisms have to have energy to carry out many of their reactions and functions. And we've recently been talking about cell transport, and so remember that many things are transported across the cell membrane using active transport. So got to have our energy, which comes for the most part from sugar molecules. Then all living things respond to their environment. We maintain homeostasis, which means we keep our conditions constant. So we're going to keep body temperature constant. We're going to keep blood pressure constant. You know, all those good things like that. And then, of course, all living things are going to evolve. All right. When we look at cells and how we figured out there were actually cells in things, we went through a number of scientists that were involved in that. So remember, Robert Hooke was the one who first came up with the idea of cells. They reminded him of jail cells because he looked at what you see over here, which is cork, and those were, of course, dead plant cells. So Hooke came up with the idea of cells by looking at cork. Here's his picture. Then we have Leeuwenhoek, who looked at the first living cells. Okay, so Hooke came up with the idea of cells. This guy looked at living cells. Then we have Sladen and Swan. So Sladen and Swan told us that organisms have cells. And so Sladen said that plants are made of cells, and Swan said that animals are made of cells. So remember, Swan is an animal, and so that's how we remember that he's the one that came up with the idea about animals. Virchow simply said that cells come from other cells. Okay, so make sure you know your scientists. Don't get them mixed up, especially Sladen and Swan. Then we looked at cell theory. All living things are made of cells. Cells are the basic units of life, and that cells come from other cells. So when we look at bodies, we know that the cell is the microcosm of the entire organism. So when we look at these levels, we said that really we start on a chemical level with our macromolecules, and then of course we get the cell, which is the basic unit of life, so smallest thing that can be alive. When you put cells that are alike, they are going to be alike, just like you see in this picture. This is one cell, and this is a whole bunch of them. If you see a whole bunch of cells put together, that's going to be a tissue. Then you put several tissues together to make an organ. So a heart is an organ. Lungs are organs. Remember we said even bones are organs because you have multiple tissues. Then you put a whole bunch of organs together and you make a system. And then, of course, you put the system together to make the organism. Okay. So we're going to go now and look at viruses. So let's pull up virus PowerPoint. And when we think about viruses, they are not living. They don't have their own cells. They have to steal a cell from something else. When they get a cell, they're going to insert their DNA into the cell and basically turn that cell into a virus factory. So this is what's going on right here. So the DNA is coming in. The DNA is going to be copied. We're going to make all these brand new viruses. They're going to fill the cell up. The cell is going to burst. Remember, the cell is going to die. The virus-infected cell always dies. So this is what our viruses are doing. Viruses are made of two basic parts, which that's not it. They're made of DNA and protein. So that's pretty much it for our viruses. We're going to zip on over to macromolecules. Macromolecules are the basic building blocks of our cells. You know, when I think about building materials for an organism, this is what I think about. If, you know, we think about what our houses are made of. Houses are made of wood and shingles and concrete and steel and things like that. Well, we're made of carbs, lipids, proteins, and nucleic acids. And remember, these molecules, they're big, and that's why we call them macromolecules. So they're great big polymers. We make them using a dehydration reaction, and then we break them down with a hydrolysis reaction. So we've been talking about this in class when we've studied osmosis, how important it is to have water inside of you because it helps you to digest. So here are macromolecules once again: carbs, lipids, proteins, and nucleic acids. You got to know your functions, and you got to know what each one is made of. And if you know those two things, you'll be able to pick out your answers quite easily. Okay, so carbs are our energy molecules. We have small carbs and then we have big carbs. So small carbs are going to be sugars, big carbs are starches. Of course, we know we can get energy from sugars a lot faster than starch. When you look at pictures of carbs, you're going to see carbon, hydrogen, and oxygen only in pretty much a 1 to 2 to 1 ratio. And it's usually your hydrogen and oxygen that are in the exact 2 to 1 ratio, but you're pretty close when you look at carbs. So just count up what you've got, and if you've got just as many carbons as you do oxygens, then that molecule is going to fall under this category. We said they give us lots of energy. They come in all sizes. So we have monosaccharides, we have disaccharides, we have polysaccharides. Okay. Now on to glycogen. Remember, glycogen is a storage polysaccharide for us. So it's what we store all of our extra sugar in, and plants are going to store it as starch. Okay. Then cellulose is what our cell walls are made of. So remember, when you look at a cell wall made of cellulose, you're looking at a plant cell. Okay. Bacteria have a cell wall. Protists have, some of them have a cell wall, but they're not going to be made of cellulose. Okay. Next category is lipids. Lipids are hydrophobic. So that's a term we've been using again this unit. Hydrophobic means they don't mix with water at all, ever. Okay. Lipids are energy storing molecules too, and they store more energy than a carb does. This is what they look like. So you've got carbon, you've got hydrogen, you've got oxygen. But look, you do not have a lot of oxygen. Okay? You have a bunch of carbon and hydrogen, and that's where all your energy is going to be stored. So look for ingredients, and if you find the right ingredients, it's going to tell you what molecule it is. So lipids have C, H, and O, but not a lot of O. Okay? And we know that examples include, our household items. We've got butter, we've got shortening, oils, things like that. Steroids fall under this category, and that is talking about anabolic steroids that people use to build their muscles up. Okay, so third macromolecule is the protein. They're the most diverse. This is where our enzymes come into play. If we look at ingredients, we have C, H, O, and N. So we add nitrogen in to the bunch here. So again, memorize what is in each molecule. Enzymes are very special proteins. They work on our chemical reactions. And if our enzymes shut down, our reactions shut down. So that means that we shut down. So enzymes are going to lower the activation energy of the reaction. So they make the reaction start faster. Okay. Don't forget they have to bind to their substrate. So this is the enzyme, this is the substrate. They fit together like locks and keys. Okay. So the enzyme has an active site on it that the substrate will fit into. Okay. Enzymes and proteins can be denatured and unfolded by changes in temperature and pH and salt. All right, last category, nucleic acids. This includes DNA and RNA. We have C, H, O, N, and P. Okay, so we've got 5 ingredients here. So make sure that you're paying attention to what the molecule is made of when you're looking for what it is. Then, of course, we've got DNA, double-stranded, and we've got RNA, which is single-stranded. Okay, here is, this PowerPoint is in Canvas too, by the way. This is a good thing for y'all to look at when studying. Okay, so moving on from macromolecules, we're going to zip over to cells, specifically cell organelles. You have prokaryotic and eukaryotic cells, right? We talked about those. Prokaryotic cells are bacteria. They don't have any organelles except for ribosomes. They have circular DNA, right? They also do have a cell wall. Don't forget that. And then we have our eukaryotic cells down here, which includes plants and animals, and it also includes funguses and protists. So you have membrane-bound organelles, okay? Plant cells and animal cells are different. Plant cells have the cell wall. They have the chloroplast here, which is where photosynthesis goes on. And then they have this thing right here, which is the vacuole, which, remember, it stores water. So that gives plant cells a lot more structure than animal cells, okay? Be sure that you know the functions of your organelles. So we divided them into several categories. Some of these organelles, they're so complex, they're only going to be found in those eukaryotic cells. So that would include lysosomes and mitochondria. So when we look at the organelles that make energy, of course, we've learned a lot about the cell membrane recently. Cell membrane, remember, is made of phospholipids and proteins pretty much. And it controls what goes in and out. There's our cell membrane. Vesicles and vacuoles. Vesicles transport, vacuoles store. They're both made of membranes. Everything has them. So we've recently talked about the contractile vacuole in the paramecium that gets rid of the extra water. And then, of course, you've got this big central vacuole in plant cells that stores a whole bunch of water and nutrients. That gives a lot of structure to that cell. So there we go. Lysosomes are only going to be in your eukaryotic cells. Remember, it says, for the most part, that lysosomes are not going to be in plant cells. So if you have to make a choice, lysosomes are not in plant cells because the vacuole does a lot of that work. But they are in higher order plant cells. But anyway, so lysosomes are going to clean up the cell. Look at that. Mitochondria makes energy. So that's the powerhouse. Remember, the chloroplasts make energy in the plant cell because they're making sugar. So that's chemical energy. Okay. Mitochondria is in both. So again, here's your plant cell. The difference is you've got chloroplast, you've got cell wall out here, and you've got vacuole. Okay, so next job of cells was to make proteins. So we're going to use the nucleus, which is control centers where the DNA is located. We use ribosomes, which are protein factories. Endoplasmic reticulum is a transport system. The rough ER has proteins attached, ribosomes attached to it, and the smooth ER doesn't. Smooth ER makes lipids. And then you have Golgi. Golgi is like your UPS store. So it will modify, it will transport, it will package, it will ship. So Golgi does a lot to the proteins. And there's our picture that's showing us protein synthesis. So again, the ER is going to transport this vesicle over here to Golgi, where Golgi is going to change it up. So it went from looking like this to this, and then the Golgi ships it off to where it's supposed to go. Okay. And then last but not least, of course, plant cells do not have centrioles, but animal cells do, and they're involved in cell division. So they look like this. There's one pair in every cell. And that is really it. So make sure you know your organelles. And now we're going to go over and look at our cell membranes and how we get across the cell membrane. So remember, cell membrane has, let's go back to this picture, lots of phospholipids, lots of proteins. Proteins are going to transport stuff for us. Okay? Things that are nonpolar, that includes oxygen and carbon dioxide, they can move right across the lipids. Okay? But if you have a polar molecule, it has got to go through a protein channel. Polar means it has a charge. So that would include glucose, and that would include water, and that would include sodium ions, potassium ions, chloride ions. So anything with a charge has got to go through this. Okay? We've been looking at diffusion, which is a form of passive transport. Diffusion is high to low. Okay? No energy is required. So we have simple diffusion, facilitated diffusion, and osmosis go from high to low and do not use energy. Okay? Active transport, on the other hand, does use energy, and you're going to be using pumps. And so anytime you see ATP, put it with active transport. So ATP goes with AT, active transport. You're going against the gradient with active transport, or up the gradient. I think the language you need to know says against the gradient. So you're going against the flow, and that's why it takes energy. So you're going from low to high. Okay? So anytime we see ATP, that's going to be active transport. We're going from low to high against the gradient. So there are all kinds of little pumps in the membranes that are going to move different kinds of molecules from low to high. Active transport is also going to include endocytosis and exocytosis. Why? Because it's big. It's transporting big stuff. When you move things that are big, you use a lot of your own energy. It kind of works the same way for this cell. You move big stuff, you are going to have to use that energy. Okay? So we've got endocytosis bringing in and exocytosis spitting out. Okay? So back over here, passive transport means we've got simple diffusion, facilitated diffusion, and osmosis. Don't forget it. Active transport, we use ATP, and we're going from low to high. Okay? Now for osmosis, which again is active transport. We're going from high water to low water. If you have something in a hypertonic solution, it's going to remove water from the cell. Hypotonic solutions are going to put water into cells. Hypertonic means more solute. It's really concentrated. Okay? Hypotonic is the opposite, less solute. So down here in this picture, we have a bunch of solute particles on this side. And so all the little yellow dots are solute particles. You've got a bunch of them here. Not a lot here. So this side is hypertonic. And water is always going to move into the hypertonic. So this is a good diagram to memorize. When you put an animal cell in a hypotonic solution, it's going to explode. A plant cell won't explode because of the cell wall. Okay? The opposite, if you put an animal cell in a hypertonic solution, so this would be like a really salty solution, it's going to pull the water out of the cell and it's going to shrivel up. Same thing for the plant cell. It's going to shrivel up. Okay? So hypotonic solutions would be like fresh water. Hypertonic solutions would be like salt water. Okay? Isotonic solution is when you're balanced. And everything is the same on both sides. Okay? So again, if you put an animal cell in a hypotonic solution, it is going to swell and burst. Okay? If something is put in a hypertonic solution, it's going to lose water. Okay? So think about when you water plants. You're going to water with fresh water, with plain water. And that water is going to go into that plant, into those cells. It's going to cause them to swell. So hypo puts water into cells. Hyper removes it. Okay? And that's really it. So on this diagram, we did this in class. Our cell has a 0.05 molar solution because we just add these two up. Our environment has a 0.03 molar solution because we added these up. So since we have more solute in the cell is hypertonic. Since we have less solute in the beaker is hypotonic, and so water is going to go into the cell. Okay, so osmosis rule is that water moves into hypertonic. okay? Go back over flashcards. If you know the basics, you will be fine on this test. It's not a hard test if you prepare for it. And it is going as a test grade, so make sure that you are thinking about that. But I would go back, look at all of the flashcards in the old modules. I would look at the old notes that I took. I would look at papers that I've given you. And go back through the PowerPoints that you saw me go through, okay? So good luck. If you have any questions, just send me an email and I'll get back to you, okay? All right, so good luck with your studying.